Today we will continue our discussion about this ILF3 in the pancreatic cancer cells. In the previous video we have discussed that the RPS18 transferred from the M1 macrophages into the pancreatic cancer cells then interact with this ILF3 or promotes the translation of this ILF3, and this ILF3 will somehow helps the pancreatic cancer cells in proliferations, migrations. So in this video we will discuss the evidence that the paper has shown that how whether this ILF3 in the upregulation of ILF3 in the pancreatic cancer cells cause lipid metabolism and other migration, upregulation of other migration properties while blocking apoptosis. So we will start with this panel A. In the panel A, what it is, they use this RPS18 knockdown short interference, this RPS18 knockdown plasmid, which is used to knock down RPS18, and they have used 3 different RPS18 knockdown plasmid, and out of this they have selected this RPS18-1, which shows the highest knockdown of this RPS18. So what they checked in this experiment, in this Western blot, is that when they use these wild type macrophages along with this RPS18, when they use only the wild type macrophages, they can see that high amount of IRF7s are produced. However, and there is a reduced amount of RPS18, since IRF7 is used to regulate RPS18, and the reduced amount of RPS18 will cause reduced amount of ILF3, and this is the control GADPH. And next they did is they knocked down this IRF7, which shows the least. Ideally you should not show any IRF7. However, since now there is no control about the translation of this RPS18, the high RPS18s are formed, and simultaneously they have seen the high ILF3, indicates that RPS18 might trigger upregulation of this ILF3. And when they use this knockout with this RPS18 knockout, they see the reduction RPS18 knocked out, and also the IRF7s are reduced. Simultaneously this ILF3 is also reduced compared to the second case, and these things are quantified in the right hand side bar plot, where we can see that when for the case of RPS18, it's highest when this IRF17 knockdown and this control interference RNA is used. And for this ILF3, the similar results are observed, that is when the IRF7 knockdown and SH control RNAs are used, then they form a highest ILF3. So previously while discussing panel A, I have mentioned about the plasmid. It is not plasmid, it is the short hairpin RNAs against RPS18 to knock down RPS18. And let's come back to this figure on the right hand side. For the IRF7, they see that in the case of wild type, the IRF7s are produced higher compared to the case of where this knockdown is used, and this is common sense, as when the IRF7 is knocked down, it is produced at a lower amount. And in the panel B, what they did is they checked the relative mRNA expressions of these 3 proteins. So in the panel A right hand side, they just quantified the protein expressions, and on the panel B they quantified the mRNA expressions, and the results are similar to the protein expressions. That is for the IRF7 knockdown and short hairpin control RNAs, they see the highest amount of RPS18 and ILF3. However, for the case of wild type exosomes, they see the high amount of IRF7 in the PANC1 cells. All these observations are done in the PANC1 cells right now. And in the panel C, they check the lipid numbers using lipid metabolites, using metabolite studies, and they are able to find out that in the case of this IRF7 knockdown and short hairpin control RNAs, they see the high amount of lipid numbers, and the cell viabilities are also higher for the same case, indicating that it's the combinations of the RPS18 It's the RPS18 that plays a role towards the cell viability and lipid numbers. Thereafter, they check this. They check the cell cycle assay using different interfering RNAs. For the control, they see that G2 and M phase are smaller. However, when they knockdown when they use the knockdown ILF3 cells, they see the high amount of G2 and S phase cells. However, when they also knock down RPS18, they see the further reduction in these G2 and S plus cells. So, and these results are they combine these results with the apoptotic assay, and the apoptotic assays are shown. Apoptotic assays are shown on the panel F. So in the panel F, what they did is they use the wild type exosomes and short interfering control RNA. They see that this is the proliferation assay. Oh, yeah, this is the proliferation assay. The tunnel assay is the apoptotic assay. So for the proliferation assay, they see that for the wild type, they see the more number of this proliferated. They see that more some number of proliferated cells. However, when the IRF7 is knocked down, they see the high proliferations of the cells. When the RPS18 is knocked down, the high proliferation is further reduced and almost comparable to the wild type. The quantifications are shown here, where we can see that for the case of IRF7 knockdown and short interfering control RNAs, they show the high positive cells, that is high proliferations are occurred. And when did the transfer assay? In the transfer assay, they are able to see that for the case of IRF17 knockdown and short hairpin control RNAs, they see that higher migrations of the cells occurs. And similarly, they did this assay, Wundhill assay. They did this wound healing assay. This quantification of this wound healing assay is shown in this panel K, where we can see that for the case of this IRF7 knockdown and short hairpin control RNAs, they see that high proliferations of the cells or wound healing rate is higher. That can be seen visually also. And for the case of migration assays, they see that migrations are higher in the case where they use IRF7 knockdown and control RNAs, not knocking down RPS18. And similar results are shown for observed for the invasions. That is, invasions are higher when the IRF7 is knocked down, but RPS18 is not knocked down. And these results are quantified in the panel L and M, where we can see that in panel M, the migrations rate of the cells are higher when the IRF7 is knocked down, but RPS18 is not knocked down. And for the M case, for the M, that is for the invasion assays in the J, which also shows the same results, that is when the IRF7 is knocked down and RPS18 is not knocked down, the invasion rates are higher. And when they do the apoptotic assay or the tunnel assay, they are able to find out that the less number of apoptotic cells are present in this IRF7 knockdown and RPS18 is not knocked down. And however, for the wild type case, their apoptotic is there, and when the RPS18 is also knocked down, they see the increased apoptotic. These results are quantified in the panel L also, which shows that apoptosis rate are higher when the RPS18 is knocked down. However, it is lower when the only the IRF7 is knocked down, but RPS18 is not knocked down. And higher for the RPS18 knockdown, along with that IRF7 is also knocked down. And these results they combine with this panel E and present that this lower apoptotic rate could be because of the high amount of this G2 plus M phase cells, and higher apoptotic rates could be because of the lower amount of this G2 and M phase cells. So these are the cells that undergo apoptosis. So from this experimental results and from the previous experimental results, they are able to make a point that in the M1 macrophages, the IRF7 controls regulates the RPS18. IRF7 regulates the RPS18, and this RPS18 will then translocate it through extracellular vesicle into the pancreatic cancer cells, where it interacts or upregulates this ILF3, and thereby helps the pancreatic cancer cells to acquire its aggressiveness properties, at the same time block the apoptosis. And if you want to understand more about these experiments in detail and want to see supplemental figures, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.